Hello, my name is Dr. Elizabeth McCready. I'm a laboratory scientist at McMaster University in Hamilton, Ontario, and with the Hamilton Regional Laboratory Medicine Program. I would like to start by thanking OGT for their support and the AMP meeting organizers for this opportunity to present our experiences with myeloid NGS gene panels. It is also important that I thank my collaborators, Dr. Darcy Butcher, Dr. Landry Nafonsum, Crystal Woodside, and Barry Ng, who have dedicated hours of hard work and time to build a myeloid NGS pipeline in our laboratory, and who continue to contribute to quality improvements to support and enhance this pipeline.
We have been providing genetic testing for the investigation of myeloid diseases for decades in our lab, initially through cytogenetic testing of bone marrow aspirates, but later by adding numerous single analyte tests, including fusion genes, NPM1, FLT3, JAK2, CALR, and MPL. Several years ago, we also developed and launched NGS pipelines for several hereditary and cancer diseases. And thus, in late 2017, when we made the decision to validate and launch a commercially available myeloid NGS gene panel, we thought that we were well prepared for the challenge. We weren't. We quickly realized that myeloid gene panel testing was not like anything else that we were doing. There was a steep learning curve related to the technical and bioinformatic nuances of myeloid NGS gene panel testing. I hope today to share with you some of our experiences by highlighting several analytical challenges of this testing through a comparison of different NGS technologies. As members of this audience are likely aware, cytogenetics has long been important for myeloid investigations by enabling evidence to support a clonal process and identifying subclones of cells that may suggest clonal evolution. Identification of genetic changes in a diagnostic sample may also be relevant for monitoring residual disease in subsequent samples. The type of genetic variant detected also have the potential to influence the differential diagnosis. We have known for a long time that there are some recurrent chromosome abnormalities that are more common in some myeloid diseases than others. The same holds true for gene variants. Most of the common genetic variants that we detect are not specific to one type of myeloid disease versus another. There are some obvious exceptions to this, though. Generally speaking, detection of a FLT3, NPM1, or a biallelic CEBPA variant is consistent with AML. Similarly, CSF3R variants are most common in chronic neutrophilic leukemia, and some specific variants like MPL are more common in some types of myeloproliferative neoplasm, such as essential thrombocythemia and myelofibrosis, but not PV. Some common genetic variants can be used to stratify MDS or AML into favorable, intermediate, and poor risk factors. For example, the presence of a translocation between chromosome 8 and 21 or the related RUNX1, RUNX1T1 fusion is often considered a favorable risk factor, unless it is also associated with a KIT mutation. TP53, AXL1, and RUNX1 mutations are also associated with poor risk stratification categories, according to the European Leukemia Network guidelines. Some of these poor risk factors are highlighted in red in this diagram and can be important to stratify risk for adverse outcomes in diagnostic samples. In blue and purple are some of the additional genes that have clinical relevance as biomarkers for either approved or pending targeted therapies, and therefore are also important considerations in gene panel design. However, mutations of any of these genes could suggest a clonal process, and this could be important in a diagnostic setting. I mention these nuances to myeloid biology to highlight their clinical utility and importance in considering them in myeloid investigations. So, which genes should we be included when we're examining these panels? In 2016, a group of hematologists, hematopathologists, and geneticists was convened within our province to consider this question and provide pathology recommendations for AML and MDS investigations that includes a recommended minimum gene list that should be examined in all suspected cases of MDS and AML. They recommended a list of 22 genes that are shown here. The genes that are checked mark single analyte gene testing already available in the lab. The rest would require a NGS gene panel to meet the minimum requirement. Using this gene list as a basis, we launched panel testing in 2018 using a commercially available myeloid NGS gene panel. Showing here are the variants detected in the first 150 sample AML or MDS samples that were assessed. We saw mutation patterns consistent with that in the literature, and there was strong concordance with single analyte testing when available. However, as we gained experience in the platform, technical considerations were raised about several targeted regions. Most notable included CEBPA, which is a technically challenging gene due to an exceptionally high GC content. We were also concerned about low coverage in several other regions, technical challenges that could impact detection of complex tandem duplications in FLT3 and KTM2A, and noise in some cases that could lead to false negatives and false positives. These are all well-established issues in myeloid gene testing that are not necessarily specific to the platform being used. As such, we sought to evaluate a second chemistry to compare analytical performance of the test with regards to some of these testing challenges. In evaluating commercially available kits, there were several criteria that we wanted to consider. First, the NGS library preparation step had to be amenable to sequencing on either the Illumina MySeq or NextSeq instrument, as these were the instruments that were available for sequencing in our lab. Secondly, the assay should align with other existing NGS workflows in the laboratory. Third, we wanted to consider the minimal DNA input requirements. This is particularly important for bone marrow aspirates with low volumes that need to be split between several labs, including flow cytometry, pathology, molecular oncology, and cytogenetics. Fourth, we wanted to look at multiplex capabilities. Our existing testing allowed for batch sizes of 8 samples, but we wanted to assess whether or not we could include more samples in a batch without compromising quality. Fifth, we asked, can the assay reliably detect relevant variants at allele frequencies of 5 to 10%? And most importantly, is there sufficient uniform coverage of all the targeted regions? We chose to compare the OGT SureSeq myeloid panel to the existing panel being used. For the genes that we are interested in testing, both tests target very similar regions of interest. This diagram shows all of the genes that we have included in our pipeline. If the full coding sequence is not targeted, then the relevant exons are indicated. The primary difference between the two panels was with regards to the targeted regions of KMT2A, which is here on chromosome 11, and I will come back to the significance of this later in the talk. With regards to technical parameters, there are some differences noted between the assays. More DNA is recommended for use with the OGT SureSeq myeloid panel, up to 1,000 nanograms, but we were able to use 100 nanograms in our evaluations successfully. The recommended batch size is also larger using the SureSeq kit, which can have substantial benefits for cost, being able to put more samples together in a single NGS run. We'll also be evaluating smaller batch sizes though too, as it isn't apparent that our sample volumes will always warrant having 12 to 16 samples on a single batch. The most significant difference between the SureSeq and existing panel is the chemistry used to enrich targets during the library preparation stage. Many of the NGS kits used in our lab and sequenced on the MySeq are based on capture hybridization chemistries, and thus we're well familiar with this type of workflow. The existing panel that we're using for myeloid is based on a PCR-based approach to enrich targets. This is not a better or worse chemistry. Both methods have theoretical advantages and disadvantages. We were interested, however, to see whether these differences in chemistry would affect the analytical performance in a myeloid setting. As I've already indicated, one of our biggest concerns is in comparing the two tests was coverage. How well does the test cover each of the loci that are targeted and at all the different nucleotides? In this graph, I've plotted average coverage at several loci in the 3 samples that were assessed using the existing amplicon-based test versus the ODT SureSeq capture hybridization. You will note that there's considerable variability in the amount of coverage at different loci using the amplicon-based test compared to the SureSeq capture. So in brown, purple, and blue is the amplicon-based, and the blue, yellow, and orange is the SureSeq. Overall, the coverage is lower using the SureSeq, and this may partially reflect the fact that there was 8 samples used per batch with the amplicon-based and 12 or 16 used with the SureSeq. But the amount of applications is relatively important across all the loci when we look at the data from the SureSeq, and that's what's really, really important, because if we the uniformity of coverage is particularly advantageous if, for some of the applications, you are concerned about copy number, for example. If you have a high amounts of variability in coverage between your different loci, being able to pick up a copy number change of 1 or 2 copies is going to be very difficult without that uniformity. Not to mention the fact that in order to get coverage, decent coverage at some of these loci, like some of these DMT1s or some of the STAG2, then we are really over-sequencing some of the other loci if you don't have that uniformity. And I've zoomed in a little bit closer on those images of just the data from the SureSeq myeloid panel. From their library preparation, you'll note that there is relatively consistent amounts of coverage between about 500 to 2,000 times coverage. There are some targeted regions that fall below 500 times, particularly in STAG2 gene region right here, which exists on the X chromosome, and it's also subject to dosage effects in males and females, so that may be part of the explanation in these particular cases. The next thing that we examined was the limit of detection using established reference controls having known variants at allele frequencies between 5 to 15%. We used the SureSeq myeloid mutation DNA mix manufactured by SureCare. Library preparations were performed using either the existing amplicon-based test or the SureSeq capture hybridization assay. Both tests performed similarly well, but some of the variants could only be seen by visual inspection of the data as they were present at lower frequencies than set by the bioinformatic pipeline. So you can see some of those variants here, but in general, particularly looking here at the OGT SureSeq, we were able to very successfully detect the variants that were present in that sample.
We also examined 61 samples using both the existing amplicon-based panel and the SureSeq capture hybridization panel. This shown here is a common recurrent SF3B1 mutation that was nicely detected in one sample by both technologies with an allele frequency of around 46%. The gene is depicted below at the bottom of the image. Exonic sequences represented by blue boxes. Some of the individual sequence reads are shown by blue and red bars, with the detected variants indicated by colored hatches. Most of these variants that were detected are background noise, and the relevant point mutation is highlighted by the arrow. The gray curves at the top of each sequence is a graphical representation of the coverage across all of the target regions, at least the targeted regions that are showing in this window. You will note relatively uniform coverage of the SF3B1 exonic regions using SureSeq compared to the other chemistry, which seems to have high coverage in some exonic segments and low coverage in others.
Here is another example of a variant detected well by both platforms. This is a relatively large exon in TAT2, exon 3, with several overlapping PCR amplicons using the existing protocol, results in a rather punctate coverage, and you can see that right here. There's strong coverage in regions where amplicons overlap, but lower coverage between these segments. This is in contrast to the SureSeq assay, which results in a more uniform coverage across the entire exon. Zooming in this bottom image, we can see a dip in the coverage for both technologies right here and here at nucleotides 484 to 485. This corresponds to a 2 base pair deletion in the sample that was present at a variant allele frequency of approximately 40%. Looking across all 61 samples, there was a high concordance between the two tests for detection of clinically relevant variants. There were some variants that were not detected in either assay, and this was generally contributed to low variant allele frequency in these particular cases. And we're investigating whether improving depths of coverage could better enable detection of these variants through our routine pipeline. We also examined other variants that were interpreted as either benign, likely benign, or variants of uncertain significance. These are highlighted in the lighter green colors. Once again, there was high concordance between the two assays with the exception of CEBPA. Several variants highlighted in orange were detected by the SureSeq, OGT SureSeq assay and by single analyte testing, but were not detectable by the amplicon-based protocol due to poor coverage. This is an example of a common CEBPA variant detected in one of the evaluation cohort specimens. It was detected by both platforms, but note the low coverage of this region in the amplicon test compared to the SureSeq coverage. Even lower coverage at this locus was noted in several of the other samples assayed using the amplicon-based technology. Conversely, consistent uniform coverage of this technically challenging GC-rich region was apparent across samples using the SureSeq assay. One of the other technically challenging variants to detect by NGS is FLT3 internal tandem duplications. These are common variants in AML and can result in duplications as small as a few nucleotides, as big as up to about 100 base pairs or more in size. 3 different FLT3 internal tandem duplication mutations are shown on this image, although we did examine others as part of the evaluation. The duplication was detected in all but one case because of a very low allele frequency. These images clearly show the duplicated segment for variants with high enough allele frequency, but a 81 base pair duplication having an allele frequency of only 4%, it was detected here, but you don't get the same jump where in the coverage rates that you do have at a variant at a little bit higher. But still, it was able to detect it using the SureSeq, and we're quite happy to have been able to see that being detected by the assay. It's very encouraging. The last thing that I wanted to point out from the SureSeq data is coverage across the KMT2A gene region. In AML, partial tandem duplications of the KMT2A gene can occur, usually involving exons 4 to 8. In the assay that we've been using recently, there were only a few of the KMT2A exons that were targeted. This, coupled with non-uniform amplification amounts, made it difficult to assess for KMT2A partial tandem duplications. These particular images that I'm showing right now are from our evaluation cohort and show the kind of an example of the types of coverage using the SureSeq chemistry in this particular gene region. There's a uniformity of coverage across all of the exons mix. It's hopeful that we will be able to detect duplications of this genomic region, but unfortunately we have not yet validated this type of variant detection, although it's very promising and we're hoping to be able to complete that validation soon. In conclusion, the assays that we compared, one was a amplicon-based assay, the other was the OGT SureSeq hybridization capture library preparation. That they were comparable assays for target enrichment at the regions of interest and had similar regions that were targeted. They also had similar minimal DNA requirements, although the recommended requirement for the OGT assay was a lot higher. We did have success using it at lower DNA concentrations. Both assays allow for batch analysis of samples, which is very good. We found that 12 to 16 assays, or 12 to 6 samples per batch with the SureSeq provided very good data, although we would also like to explore what the effect of having fewer samples in a batch would mean as far as coverage goes. Improved uniformity of coverage using the captor hybridization library preparation protocol compared to the amplicon-based technology was by far one of the greatest surprises. Well, it wasn't a surprise, but one of the greatest encouraging things that we found with this evaluation. And we really liked that coverage that was uniform. And I think it's going to provide much better quality analysis across all of the regions that we're assessing, as well as it was also cleaner data with fewer background noise. The detection of variants at allele frequencies of 5% or greater was also very good using both technologies, assuming that there's sufficient coverage. So that is also encouraging. And we're looking forward to using this SureSeq panel for more analyses as we move forward with this test in our lab. Thank you for your time.